Hello again everyone, thank you very much for joining us again. So we've talked through in this series of videos about the importance of neutrophil extracellular traps and how they are pivotal to our immune response and how bugs and bacteria can manipulate them. And we've talked about the individual components of the immune response too. For Volition as a company and to help move research forward and improve diagnostic enrichment and prognostication and lead to personalised medicine, we have spent a lot of time working on developing a model of NETosis that's clinically relevant. And what I'm going to do in the next few slides is take you through the models that we've developed. This is really important to help us understand neutrophil biology, to understand the ways in which nets are triggered and nets are released. We hope to link that clinically to studies showing patterns of net release and how that influences patient outcomes, and ultimately identify the molecular pathways at the very, very heart of this process. Because if we can identify the molecular pathways, we will be able to identify drugs and ways of attenuating, moving them and changing them. And you can see how we can develop therapeutic strategies for patients. Developing model systems for studying sepsis. So there are various ways we can do it. Here on this slide you can see more of those purple top tubes, K2 EDTA tubes. And we can either study isolated neutrophils, which scientifically is quite a nice thing to do because you know you're just working on a neutrophil, or we can study neutrophils in whole blood, which is a lot trickier. The advantages of isolated neutrophils is you're just stimulating the neutrophil, and so you can just see what you're using. However, you can see from all the models, that amazing slide with platelets, neutrophils, complement, monocytes, etc., that actually neutrophils absolutely don't work in isolation. They work together with all these other factors that you can see in the bottom of the slide here. So we've developed a model for working on neutrophils in whole blood, which we think is much more biologically relevant. And if we think back to that slide where I showed the 3 different types of NETosis, perhaps you start to see most of the work and research has been done in isolated neutrophils. And it's like you have to hit isolated neutrophils harder to stimulate them. And perhaps an artifact we're seeing in some of the earlier research is that we stimulated, or the triggers that we use to stimulate neutrophils are just too hard. And that might be slightly distorting some of our scientific findings. Other issues. An awful lot of work is done in mice, but mice don't perfectly represent human biology. A bit bigger, we're more complicated, and we have a much more complicated immune response. A very physical problem with doing experiments and practicals in mice is that they are little and that you have a small amount of blood to work with. And so that can really limit what you're physically just able to do. Mice also only have about 10, maybe 20% of the neutrophils that humans do. And so a human neutrophil model of sepsis, or a model of neutrophil sepsis in humans, is something we've been chasing or developing with our innovation lab, and you can see why it's not equivalent to a mouse model of neutrophil activation and sepsis. So over the next series of talks, I'm going to cover net release in isolated neutrophils and then net release in whole blood. I'm going to try and explain those mechanisms to you and the work that our innovation lab have been doing.